Hey guys, welcome back to my channel. This is Dr. Rajeshwar from yrpharmatube. In the previous lecture, we have learnt the various roles played by the drug metabolism. If you did not watch the video, click on the I button on top right of this video to watch it. In this video, we shall try to understand the classification of drug metabolism. At the end of this video, you should be able to list the various chemical reactions involved in drug metabolism or biotransformations. To define phase 1, phase 2 and phase 3 reactions and the respective roles of the three phases of drug metabolism. When the drug is introduced into the human body, it is metabolized and transformed partly or wholly into the other substances, which are called the metabolites. These biotransformations are catalyzed by enzymes which are found mainly in the liver, although they are also found in lesser quantities in other tissues such as intestine, kidney, and lungs. From the point of view of drugs fate in the human body, drugs can be divided broadly into three types. Number 1, drugs which undergo enzyme catalyzed reactions. Number 2, drugs which are not metabolized and are excreted unchanged. And number 3, drugs which undergo spontaneous reactions when given the appropriate physical conditions such as pH, temperature, or when they contact a suitable physiological compound with which they can react. The majority of the drugs belong to the first type, but some of the drugs can undergo a combination of enzyme catalyzed reactions and spontaneous reactions. Although enzyme catalyzed reactions are carried out by the tissue of the body, it is now becoming clear that some transformations of drugs can also be carried out by the gut or intestinal flora. The drug or the xenobiotic metabolic reactions or the phases of detoxification are divided into three phases. Number 1, phase 1 reactions. Number 2, phase 2 reactions. And number 3, phase 3 reactions. It is important that these pathways are studied as the root of the metabolism of a drug, which can determine whether the drug has any pharmacological or toxicological activity. Phase 1 reactions. The phase 1 reactions are thought to act as a preparation of the drug for the phase 2 reactions. That is, phase 1 functionalizes or modifies the drug molecule by producing or uncovering a chemically reactive group on which the phase 2 reactions can occur. Thus, the phase 1 reactions are called either functionalization or modification reactions. All the phase 1 reactions involve the conversion or the biotransformation of a drug or a xenobiotic to a more polar or water-soluble metabolite, and so the drug can be excreted more easily than the parent drug from the human body. Generally, this conversion is seen in the drug molecules containing the hydroxyl, amino, thiol group, etc. Thus, these phase 1 reactions largely depend on the hydrocarbon skeleton functional groups present in the drug molecule. This biotransformation can lead to either activation or inactivation of the drug. This results in the loss of pharmacological activity. However, active and chemically reactive intermediates may also be generated. Phase 1 reactions are especially important in prodrug metabolism. Prodrugs are pharmacologically inactive derivatives of the active molecule that are designed to break down within the body to release the active drug, which means that prodrugs are pharmacologically inactive compounds in vitro, that is outside the human body, but they are rapidly converted to biologically active metabolites, often by hydrolysis of an ester or amide linkage. The prodrug approach is often used in pharmacy to overcome problems such as poor absorption, instability, or toxicity when the parent drug is given orally. Examples include anti-cancer agents such as cyclophosphamide or antibiotics such as pivmecillinum. The prodrug approach can also be used for several other purposes with respect to the patient compliance. We will learn more about prodrugs in the future lecture videos. In rare instances, phase 1 metabolic reactions are associated with an altered pharmacological activity. In these phase 1 metabolic reactions, a variety of enzymes act to introduce reactive and polar groups to the molecules. Therefore, these phase 1 reactions or enzymatic reactions are sometimes called enzyme-dependent reactions. These enzyme complexes act to incorporate an oxygen atom into non-activated hydrocarbons, which can result in either the introduction of hydroxyl groups or nitrogen, oxygen, and S-dealkylation of the drugs. The reaction mechanism of the cytochrome P450 oxidases proceeds through the reduction of the cytochrome-bound oxygen and the generation of a highly reactive oxyferryl species according to the scheme as given here. Phase 1 reactions are non-synthetic reactions and are typically divided into 5 types. Number 1, oxidation. Number 2, reduction. Number 3, hydrolysis. Number 4, hydration. And number 5, rarer and miscellaneous phase 1 reactions. The oxidation reactions are classified into 2 categories. Number 1, the oxidations involving or catalyzed by cytochrome P450 enzyme, that is, the microsomal mixed function oxidase system. And number 2, the oxidations not catalyzed by cytochrome P450 enzyme, that is, the oxidations which do not use the microsomal mixed function oxidase system. The oxidations involving cytochrome P450 enzyme involve several reactions such as aromatic hydroxylation, aliphatic hydroxylation, epoxidation, oxidation of benzylic carbon atoms, oxidation at allylic carbon atoms, oxidation at carbon atoms alpha to the carbonyl and imine groups, oxidation involving carbon heteroatoms, oxidative deamination, oxidative decarboxylation, phosphothionate oxidation, and lastly, dehalogenation. The oxidations not catalyzed by cytochrome P450 enzyme, but the reactions oxidized by the enzymes such as alcohol dehydrogenase, aldehyde dehydrogenase, xanthine oxidase, amine oxidase, aromatase, and alkyl hydrazine oxidase enzymes. The second type of phase 1 reactions are the reduction reactions. These reduction reactions are of 5 types, which include the reduction of carbonyl compounds, that is aldehydes and ketones, reduction of nitro and azo compounds, azido reductions, reduction of sulfur-containing compounds, and reductive dehalogenation. The third type of phase 1 reactions are the hydrolysis reactions. These hydrolysis reactions include ester hydrolysis, amide hydrolysis, and hydrazide and carbamate hydrolysis. The fourth type of phase 1 reactions are the hydration reactions. And the last and fifth type of phase 1 reactions are the miscellaneous reactions. All the phase 1 reactions can result in the activation of a drug, the conversion of an initially inactive compound into an active drug, and the conversion of an active drug into another active drug. If the metabolites of phase 1 reactions are sufficiently polar, they may be readily excreted at this point. However, as explained above, many phase 1 products are not eliminated rapidly and undergo a subsequent reaction in which an endogenous substrate combines with the newly incorporated functional group to form a highly polar conjugate. Phase 2 reactions or the conjugation reactions. In subsequent phase 2 reactions, the activated drugs or xenobiotic metabolites from the phase 1 reactions are conjugated with charged species such as glutathione, sulfate, glycine, or glucuronic acid, and thus these phase 2 reactions are called the conjugation reactions. Conjugation occurs mainly in the drugs containing carboxyl, hydroxyl, amino, and thiol groups. These reactions lead to the formation of a covalent bond between the functional group of the drug molecule or on a phase 1 metabolite with endogenously derived compounds as mentioned above, that is glucuronic acid, sulfate, glutathione, amino acids, acetate, etc. The addition of these groups onto the drug molecules produces the products in the conjugation reactions have increased molecular weight and tend to be less active than their parent molecule. These highly polar conjugates are generally inactive and are excreted rapidly in the urine and feces. Thus, the phase 2 reactions are usually the true detoxification pathways and give products that account for the bulk of the inactive excreted products of the drug molecule. This is comparable with the phase 1 reactions, which often produce active metabolites. The addition of large anionic groups such as glutathione detoxifies reactive electrophiles and produces more polar metabolites that cannot diffuse across membranes and may therefore be actively transported. These reactions are catalyzed by a large group of enzymes called the transferases, which in combination can metabolize almost any hydrophobic compound that contains nucleophilic or electrophilic groups. One of the most important classes of this group of the glutathione S transferases. Many of the reactions of both phase 1 and phase 2 reactions are capable of being performed on the same compound, and thus there is a possibility of interaction with the various metabolic routes in terms of competing reactions for the same drug or the substrate molecule. The table given here gives the information of the type of conjugation reaction, various enzymes involved, the cofactors, and the location of enzymes utilized in phase 2 reactions. Phase 2 reactions are divided into several types, which include conjugation with sugars, which are further classified into glucuronidation, that is, the conjugation with glucuronic acid, which may be of O, N, S, and C glucuronides, and conjugation with other sugars. Sulfate conjugation, methyl conjugation, acetyl conjugation, amino acid conjugation, glutathione conjugation, conjugation with coenzyme A, fatty acid and cholesterol ester conjugation, cyanide conjugation, and condensation reactions. Lastly, the phase 3 reactions. After phase 2 reactions, the drug conjugates may be further metabolized and excreted from the human body, which means that in phase 3 metabolism, the conjugates and metabolites of phase 2 reactions are excreted from the cells. A common example is the processing of glutathione conjugates to acetylcysteine conjugates. Here, the gamma glutamate and glycine residues in the glutathione molecule are removed by the gamma glutamyl transpeptidase and dipeptidase enzymes. In the final step, the cysteine residue in the conjugate is acetylated. Conjugates and their metabolites can be excreted from the cells in phase 3 of their metabolism, with the anionic groups acting as affinity tags for the variety of membrane transporters of the multi-drug resistant protein family. These proteins are members of the family of ATP binding cassette transporters and can catalyze the ATP dependent transport of a huge variety of hydrophobic anions and thus act to remove phase 2 products to the extracellular medium where they may be further metabolized or excreted. The reactions involved in all these phases, in particular the phase 1 and phase 2 reactions, will be discussed in terms of reaction type and the enzyme involved in the next lectures. This is the list of references followed for the lecture. That's all in this video, the classification of drug metabolism. In the next lecture, we will discuss about the phase 1 oxidation reactions. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.